Hi, I'm Allison Vanisko, and my partner Judy and I are here to tell you about Meiosis 1 and Meiosis 2 through an instructional video. First, we're going to start off with some vocab so we better understand the topic. Our first vocab is, what exactly is meiosis? Meiosis is the division of a single diploid cell into 4 haploid daughter gametes. Meiosis and cytokinesis produce haploid gametes from diploid cells and the reproductive organs of the parents. The next vocab is, what exactly is a diploid cell? A diploid is a cell that has 2 sets of homologous chromosomes. For humans, that is 23 from mom and 23 from dad. Next is, what is a haploid gamete? A haploid gamete is not a full cell because it only contains 1 set of chromosomes. For humans, that's chromosomes 1 from 23 from either mom or dad. Haploid gametes are the egg and the sperm. What are homologous chromosomes? They are the 2 chromosomes that make up a matched pair in a diploid cell. The mother gives a set of chromosomes, 1 through 23 for humans, and so does dad. The chromosomes then pair up, making them homologous. So chromosomes 1 and 1 pair up, and so on and so forth. What is a sister chromatid? It is one of the 2 identical parts of a duplicated chromosome in a eukaryotic cell. Prior to mitosis, sister chromatids remain attached at the centromere. What is the centromere? The region of a duplicated chromosome where 2 sister chromatids are joined and where the kinetochores attach. What are microtubule spindles? They are strings that come out of the centrosome that create what looks like a latitude line and then pull the homologous chromosomes apart into opposite ends of the cell. What are centrosomes? Material in the cytoplasm that create microtubule spindles during meiosis and mitosis. What are centrioles? They are a structure in an animal cell composed of cylinders of microtubule triplets. An animal usually has a centrosome or the pair of centrioles involved in cell division. What is a nuclear envelope? A nuclear envelope is a double membrane that encloses the nucleus. Finally, what is a kinetochore? They are where the microtubule spindles attach. Be careful not to get kinetochore and centromere confused. Now that we understand the vocab involved in meiosis, we're going to start going through each stage step by step. First off is interphase. Before a diploid cell launches into meiosis 1, there is a prepping phase called interphase. In interphase, the chromosomes duplicate, 2 genetically identical sister chromatids are attached together, and the centrosome also duplicates. Once all of this happens, interphase is done and it is on to prophase 1. Prophase 1. In meiosis, there are 2 separate stages, or phases more or less. First off is prophase 1. In prophase 1, there's a process called synapsis that takes place. In this process, homologous chromosomes come together as pairs. This results in 4 chromatids called a tetrad. Also during synapsis, the chromatids of homologous chromosomes exchange genetic segments of genetic information in a mini process called crossing over. This crossing over creates genetic variability. Also in prophase 1, the chromosomes condense, the nuclear envelope breaks into fragments, the centrosome goes to opposite poles, and the centrosomes encapture the tetrads in the microtubule spindles. Metaphase 1. In metaphase 1, the chromosome tetrads align on the metaphase plate, which is at the equator of the cell. The sister chromatids are still attached by their centromeres. The spindle microtubules from one pole of the cell attach to one of the homologous chromosomes of the tetrad by the kinetochore at the centromere, and the spindle microtubules attach to the other homologous chromosome of the tetrad by the other kinetochore. This phase makes it easier for the cell to go throughout the next phase, anaphase 1. Anaphase 1. In anaphase 1, the tetrads split into 2, but still leave their sister chromatids all attached by the centromeres. However, genetic information has been exchanged by the process of crossing over, so the sister chromatids do not look the same as when they first formed. Next, the spindle microtubules start to rear their sister chromatids to their half of the cell. Telophase 1 and cytokinesis. In telophase 1, the chromosomes arrive at the poles of the cell and have a complete haploid set. However, they are still duplicated, and each chromosome still consists of 2 sister chromatids. In cytokinesis 1, the chromosome begins to uncoil and the nuclear envelope reforms. This preps the cell for the next phase, meiosis II. Now we're on to meiosis II. Now that the first part of meiosis is covered, it is time to explore the second half. Meiosis I is very similar to mitosis, but meiosis II is a bit different because the daughter cells split again to make 4 haploid daughter gametes. Prophase II. In prophase II, a spindle forms and moves the chromosomes towards the middle of the cell. In metaphase II, the chromosomes align themselves in the metaphase plate, which is at the equator of the cell, with the kinetochores of the sister chromatids of each chromosome facing opposite poles of the cell. Anaphase II. In anaphase II, the centromeres of the sister chromatids separate, and this makes individual daughter chromosomes that move towards opposite poles. Telophase II. In telophase II, nuclei form at the cell poles. The final step of meiosis, cytokinesis. Cytokinesis II then separates the cytoplasm, resulting in 4 daughter cells with the haploid number of chromosomes. Remember, the haploid number is half the amount of chromosomes a cell needs to function. For humans, this is 23. Thanks for watching our instructional video about meiosis. I hope you have a better understanding of vocab and what each phase entails. Thanks so much!